In this video, we're going to look at how we use DNA fingerprinting and gel electrophoresis to run a DNA fingerprint to be able to compare DNA. So here's a sample of some DNA that might be used at a crime scene to analyze the DNA evidence and to solve a crime. Remember, first thing you have is you've got to use a restriction enzyme because all human DNA is 46 chromosomes, and so if you were to look at them, they would all look the same. But we all have very many unique sequences in those 3 billion base pairs, and they're going to get cut differently. And if we use different restriction enzymes, they're all going to get cut in unique ways, and that's going to mean that we all will have unique cut pieces of DNA. But that's not going to help. We've got to now separate them out, so we can use gel electrophoresis to separate them out by size. So let's look at how that works. So if we take this DNA and we digest it with the restriction enzymes, and then we load it into wells, DNA is negatively charged. So we can pull it through a gel, and it'll get pulled towards a positive end if we run an electrical current through the gel. And DNA, because it's all cut in all these different sizes, all the pieces don't move the same. Small pieces are going to move through that gel very quickly. Large pieces are going to move much more slowly. So what we're going to see is, as the DNA moves through the gel, these banding patterns are going to appear. And the bands, again, separate by size. Smaller pieces moving faster, bigger pieces moving slower. But because we all have many, many different unique pieces when we cut our DNA up, we all have unique banding patterns. And that's going to give us all a unique DNA signature. It's much like a barcode when you buy something at the supermarket. All those bands on the bar, a computer can read that and know exactly what it means. And we can read our DNA in a very similar way with these unique banding patterns. So here's what it actually looks like in the gel. And they put a dye on the DNA and it causes it to phosphoresce and glow. And then you can compare those DNA sequences and see if you've got similarities or differences. And that's it. Very short video. Good day.